we have started this lecture series who are interested to deliver and have expertise in different domains. So we have started to disseminate knowledge to the needy people and who are interested to learn and who are interested to deliver. That is purpose. So this is free lecture series and we will continue it until we will not get expert people. Randomly we cannot take random people. If they have some expertise, they have very good background, then we allow them to deliver. And so I am acting as a mediator. So in this way that knowledge will percolate, disseminate rapidly to the society. That is my purpose. So with this note, I welcome today's speaker, Dr. Ashwini Kumar, and he did his PhD from NIT Raipur, and then he joined IIT Delhi as a postdoc fellow. He has published several research papers. He is a reviewer of several journals also. He has published more than 10 research paper and review paper and book chapter also, several book chapter also. Nowadays, he is working at Nimrana. Just let me check his institute name. You all have his institute name, I think, but still. Sometimes it's difficult to remember. So he has published 22 journal articles, and 9 book chapters and books. He has been associated editor for a special issue in Frontiers in Medical Technology and regular reviewer of Material and Design, Elsevier. He has delivered talk in various national events and been a jury member of 2 consecutive years for Darwin, an international conference organized by Sumaya Trust. With this brief note, I request now Dr. Ashwini ji to deliver his lecture, and I will allow participants to come join because more than 107 participants have registered. But you can see here that's why I limit these things. Who will attend? They will only get certificate, and they must interact. That's why recording is not available, because everybody claims that they will watch recorded, but they don't watch. So I restrict these things. If you are attending, then you will get certificate. Not always. Here also we have not decided yet whether you will get certificate or not. But you must interact. You must ask question. This is not one way. That's why we have written that interactive session. So no one is in mute condition. You can raise hand and you can ask question in between also. But I prefer that you ask question after finishing. But if you have any doubt, you may ask. So thank you, Ashwini ji, for sparing your time. And obviously, your contribution will be remembered by society, because we all should contribute for upliftment of science, and you are also contributing. So thank you.
Thank you so much, sir, for starting with me this beautiful session series. As all of you know, today we'll be discussing something on tissue engineering, the science of it, and the hope that has arisen because of this particular field of research. So I'll just start presenting. Sir, is this visible?
Yes, it's visible.
Okay. So I'm Ashwini Kumar. I work as assistant professor in biotechnology and bioinformatics area at NIIT University, Nimrana, Rajasthan. It's just at the border of Haryana and Rajasthan. It is in Alwar district, and it is the first subdivision that you will encounter as you proceed from Gurgaon towards Rajasthan. Okay. So we all have heard a term called organ transplantation. So organ transplantation is a surgical technique whereby a terminally diseased tissue or organ is replaced by a functional tissue or organ, either harvested from a live donor or from a cadaver. Cadaver is the scientific name for dead body. So it is done when a tissue or organ completely stops working, and the transplantation remains the final alternative so that the person can survive. So it is a surgical replacement of a damaged or dysfunctional organ and tissue by a functional organ and tissue. Currently, we can have the tissue or the organ from the live donor. The live donor can be the patient itself, and in that case, if a tissue is harvested from the patient itself, it's called autograft. If a tissue is or a organ is harvested from a person from the same species, another person of the same species, it's called allograft. And currently, it is the most used technique for organ transplantation. And we can also have certain tissue and organ from the cadaver. It is also called allograft. There's another term in grafting which is called xenograft, whereby a tissue or organ is harvested from a organism of a different species. So, if suppose we are transplanting a heart from a monkey to a human being, that will be called a xenograft. Though it has this organ transplantation, the conventional form of organ transplantation is there for a long period of time, but still we have a lot of drawbacks associated with it, or challenges associated with it. Firstly, looking at the live donation, we have very less amount of tissues that can be harvested from a live donor. So if I may ask the participants, especially the students who are participating, what tissues or organs can you harvest from a live person? You can just unmute and reply.
Tumors?
Sorry, pardon?
Sir, tumors.
No, we don't transplant tumors. We only want to transplant a good functional organ or a tissue.
Liver?
So what tissue can
Kidney?
we take? Okay, out of two, we can, yes, take one kidney.
Poonam madam, please mute your mic. Poonam madam.
What other organs and tissue can we take from a live person and be transplanted to another person in need, apart from kidney?
Lung?
No, we can do with single lung, but we cannot do with single kidney. We cannot do with single lung. We cannot also
Liver?
harvest. We can take a part of liver, yes. No, again, we cannot take heart from a live person. He or she will die. Okay, so we can have single kidney out of the two. In case a person so there are developmental condition where a person only develops a single kidney. In that case, that kidney cannot be harvested from that person. So out of two, one kidney can be taken for transplantation to another person. A part of liver can be taken, not the whole liver, because liver has got the most regeneration capacity, and therefore only a small part does all the work. If it is provided at the specific place, the whole liver will be grown. We cannot take out lungs, we cannot take out heart. We can have some part of the bone, we can have some part of the skin from the live donor. From the cadaver, there are more options, but there are again different types of challenges, because the first crucial thing that is detrimental while deciding whether an organ or tissue can be harvested from a dead body or not is the time of death. Because depending on time, different tissues and different organs start deteriorating or decaying. So time of death is very crucial, and that decides, based on the time it is decided, which organ can be harvested or not. So from a cadaver, we can take out kidney, we can harvest lungs, heart, bone, skin, cornea, eyes. There are a lot of things. But still, apart from this time of death challenge, other challenge is the risk of transmission of diseases. Though there are a lot of investigations done before a cadaveric organ or tissue is transplanted, so that the risk of transmission of disease can be minimized, but still, technically, that is a risk that is there with the cadaveric donation. If you can see the graphs on both the sides, left and right, you can see. So these are taken from organindia.org and noto.gov.in. Around 2 lakh cornea donations are needed annually in India. Out of that, only 50,000 are available on an annual basis. There are a lot of organs. There are kidney, heart, livers required. But availability is, again, you can see, if you can just focus on the minute written material, there are around 2 lakh kidney required, but there are around 1,684 kidneys available for that requirement. On the right hand side in the bar graph, you can also see till the data is till 2021. The The longer line is for kidney, the dark green is for the liver, and the red one is for the heart. You can see obviously heart transplantation is a more technically difficult task, and fortunately, not many people require that. But there is a lot of demand for kidney. The first is kidney, and then the liver. But the problem is there is a lot of gap between the number of donors available and the number of recipients. And therefore, many recipients who are terminally ill, they die without having any chance to get a transplant. And therefore, this field of research, tissue engineering, came into existence to fill up that gap. If we can design, we can fabricate a complete functional organ or tissue in the lab, we won't be requiring any live or cadaveric donor. So that was the background why tissue engineering came into place. Now, the term tissue engineering was officially coined at National Science Foundation workshop in 1988. Just a few years after that foundation workshop, in 1991, Henken and Scalak gave the first definition of tissue engineering, and then in 1993, the definition was just a bit modified by two scientists, Langer and Beckenty. And currently, this is the most widely used definition for tissue engineering, that states that tissue engineering is an interdisciplinary field of research that applies both the principle of engineering and the process and phenomena of life sciences towards the development of biological substitutes that can restore or maintain or improve the function of a diseased or damaged tissue. So we apply a lot of things, right from the basic engineering knowledge to the core knowledge of biological sciences. These are clubbed together so that a new tissue can be fabricated completely in the lab. But again, there are so this process of fabricating a complete new tissue was also brought into concept because apart from the challenge of the gap between the donor and recipient and the risk of transmission of disease, there were other technical challenges that were very costly for the patient. So technically, the person who is undergoing a transplant, he or she is administered a lot of immunosuppressive drugs before and after the protocol, so that before it is done, so immunosuppressive drugs are the medicines that suppresses the immune system. You might have heard that there are a lot of cases where the transplanted tissues are rejected by the body. So this rejection is actually an immunological phenomenon. So because body can perceive something as non-self or as a foreign, so the tissue or organ that is transplanted into a patient, most of the time, or almost 100% of the time, the body of the recipient try to reject that transplant, the tissue or the organ. And to minimize the risk of rejection, immunosuppressive drugs are given before the procedure starts and after the procedure is finished. And post-procedure, the immunosuppressive drugs are administered for almost the lifetime, so that the body should not reject the transplant at any given point of the time. So, believe me, these immunosuppressive drugs are actually very costly. So, it costs a fortune for the patient, and therefore, it is very, very mentally tasking for the patient to have a graft. So, if we could provide an alternative to these live or cadaveric donor through a completely engineered tissue, a lot of these drawbacks and challenges can be overcome. I hope you all know that any tissue in the body is basically can be divided into two parts. One is parenchyma, that is the functional part, and the extracellular matrix that gives the support, that function as a support structure or gives the dimension to the tissue. So every organ is almost of the same size in different individual. So a kidney will be of the same size in my body, in your all of your body. Similarly for the heart, for lungs. So this dimensional aspect is given by extracellular matrix, and the functional part is given by the parenchyma. If you can see this picture, you can see in the A part is the normal adipose tissue or the fat, and the just adjacent to that you can see the white portion, the white picture. This is only the ECM, the extracellular matrix of the fat, where the cells have been removed. Only the ECM is now left. You can below you can see the scanning electron microscope picture of both the sections. On the left hand side, you can see it is more dense because of the presence of the fat cells or adipocytes. The right hand side SEM picture is more porous in appearance because only ECM is there. Okay, so I use this analogy every time whenever I try to explain the concept of tissue engineering. I hope you all have seen such kind of structure around you. So it is the beginning of constructing a building. Here you can see a lot of bricks, iron rods, cement, soil, and different items are used. They are combined to give this particular structure. But this is a very preliminary structure, and it is not fit to live. Then after a few more modification, the structure changes something to this. Again, this is not good to live, to stay, but it looks better than the previous one. After more modifications like giving colors and aesthetic attachments, we can have a building where people can live actually. So this is an analogy where you can think the fabrication of a tissue right from mixing all the ingredients. So these are three elements, basic elements in tissue engineering. We require certain biomaterials, and these biomaterials or biopolymers are used to fabricate the extracellular matrix or the synthetic extracellular matrix, which is called a scaffold. Scaffold in Hindi is called dhacha. Then we require the cells that will give rise to the parenchyma or the functional part of the tissue, and we require certain protein and non-protein molecules such as growth factors and other metallic elements, which help in the cellular growth, proliferation, differentiation, and cell-to-cell communication. So very basically, we can have the cells isolated from the patient itself. We can expand or proliferate the cell in the lab. We can create a three-dimensional scaffold which mimics the extracellular matrix. We can combine the proliferated cells and the extracellular matrix so that a new tissue can be given, rise in the lab. And after checking for all the required function and all non-functional aspect, the tissue can be implanted back. Okay. Again, here you can see a term implantation or implant. Implantation comes from implant. Can anyone again tell me the difference between transplantation and implantation? We often use these two terms very interchangeably, but there's a technical difference between these two. Anyone, any idea that comes to your mind? What is the difference between transplantation and implantation?
Sir, implantation may be included from one's body and transplantation is from other person's body.
Okay. Yeah, one of the points. Any other point? Okay. No issue, not taking much of.
Implantations is
Yes, please.
used for tissues or cell. Transplantation is for organs.
Okay, not exactly. So whenever we shift organ or tissue from one person to another, it is transplantation. Like transfer means changing a place of work. So transplantation is changing the place of that particular tissue or organ from one person to another. When we put something inside a body that is completely synthetic, that is called implantation. So a metallic we normally use the word implant with metallic implant. So you might have heard of knee implant or hip implant. So they are implantation, they are not transplantation. But heart transplantation is a transplantation, or kidney transplantation is a transplantation because we were placing kidney from one person to another. So that's the technical difference. Whenever something is completely synthetic, it is implantation. Whenever a natural tissue is transferred from one person to another, one body to another, that is transplantation. So in the last slide, we were talking about the three elements of tissue engineering. First is the scaffold. Unless until we have, so if you can recall the building transition, unless we have all the ingredients which gives the dimensional aspect to a building, we cannot proceed to a livable building. And that's why the first thing that we require to understand is the scaffold that mimics the extracellular matrix. Any material, any biopolymer that we choose to fabricate the scaffold for a particular tissue or organ must be biocompatible. Biocompatible means it should not cause any undesired effect when present inside the body. It should be degradable if the degradation of the polymer is required, and it should not be non-immunogenic. When we say about non-immunogenic, that means the scaffold as a whole should not elicit any immunogenic reaction. And if it is degradable polymer, the degradation product should also be non-immunogenic. ऐसा नहीं हो कि the scaffold as a whole is non-immunogenic, but the degradation products are immunogenic. That won't solve the purpose. The second quality of a good scaffold is uniform porosity. Why do we need porosity? Then all the natural extracellular matrix are also porous. We require this porosity because unless until a scaffold or ECM is porous, there won't be cell-to-cell communication, cellular communication or cellular signaling. Cellular signaling is not only about the movement of protein and non-protein molecules so that the cell can proliferate. It is also about the transfer of nutrients. And that's why porosity of a scaffold is very, uniform porosity is highly required. The scaffold should be mechanically stable. That means the mechanical and physical property of the scaffold should mimic that of the tissue that we require to replace. So it should be mechanically stable. ऐसा नहीं हो कि while implanting it gets torn very easily or is very labile towards any physical damage. So we need to check a lot of mechanical properties before we proceed to another things. The biopolymer that we require to prepare a scaffold can be natural or synthetic. If you have heard of polymers like cellulose or alginate or chitosan, these are all natural. If you have heard of acrylic acid or polylactic acid, these are completely synthetic polymers. And therefore, after all these properties of biocompatibility, degradability, porosity, and mechanical stability, the scaffold should also show sufficient cell adhesion because cell require that space to adhere and then the cell will proliferate and differentiate. It should promote the cell differentiation if the differentiation is required. Being porous, it should also promote cell migration, cellular signaling, and sufficient cellular metabolism. The second element of a good tissue engineering product is cells. That performs the role of the parenchyma part of the tissue. So the cells can be a terminally differentiated cell. There are a lot of agencies or organizations from where you can purchase the cells. Either they can be terminally differentiated. So when we talk of terminally differentiated, you might have heard of a term called progenitor cell. So a progenitor cell is just a precursor cell. That means a stage just before a completely differentiated cell. So they are called terminally differentiated. You can use those cells, but normally we don't use a terminally differentiated cell for a translational purpose because they have a limited lifespan. And that's why the most used type of cells are either a mesenchymal stem cell or induced pluripotent stem cell. I hope many of you have heard of induced pluripotent stem cell.
Yes, sir.
Okay, great. So pluripotent stem cell was so this different classification is based on the potency of differentiation. So when we talk of potency, a cell can be totipotent, pluripotent, multipotent, or oligopotent. A totipotent cell is only the zygote. Apart from zygote, there is no cell in the body that is totipotent, that have the total potential of giving rise to an organism. Moving on from zygote, we have embryonic stem cell that is present in the blastula stage of the embryonic development. They are called pluripotent and not totipotent because except for the trophoblast layer of the placenta, an embryonic stem cell can give rise to all the cells present in the body. They can be differentiated to all the cell except for the trophoblast cell, and that's why they are not totipotent. Moving again from pluripotent to multipotent, there are certain sections of the cell or there are certain cells found in different specific locations that can give rise to a particular lineage of tissue. That means if you have heard of the Hindi word khandan, so they can give rise to a particular lineage, particular khandan of the cells. So when we talk of mesenchymal stem cell, they can be differentiated only into bone cell, osteoblast, cartilage cells, chondroblast, adipocytes, fat cells, and a skeletal muscle cell. Similarly, hematopoietic stem cell can only be differentiated into all the blood cells. And that's why hematopoietic stem cells found in the bone marrow, mesenchymal stem cells found in these locations are multipotent stem cell, while all the progenitor cells are oligopotent. That means they can only give rise to their terminally differentiated cells. Now, Professor Shinya Yamanaka from Japan he received Nobel Prize, if I'm not wrong, in 2006, and his work was that he transformed a terminally differentiated skin cell into a cell that mimicked the embryonic stem cell. Now, one of the basic motive of doing this was when the research on embryonic stem cell was initiated, the only source of harvesting embryonic stem cell was to destroy the embryo. Though the embryo was not implanted in the uterus, they were mostly the discarded embryo after the IVF procedure. But still, there were a lot of ethical committees that came up, and very stringent guidelines came out, where it was almost prohibited not to destroy an embryo in order to harvest the embryonic stem cell. And because embryonic stem cell holds a lot of potential in terms of differentiation, the team led by Shinya Yamanaka from Japan, they put back a terminally differentiated skin cell into embryonic stem cell-like characteristic, and from that stage, that embryonic stem cell-like cells could be differentiated into all the cells of the human body. And because this property was induced, these cells are called induced pluripotent stem cells. And currently, all the labs which are actively working on tissue engineering, they are either working with mesenchymal stem cells or with iPSCs. One advantage of getting MSC or iPSC is that they can be autologous. That means they can be harvested from the patient itself, and therefore they hardly have any issue of getting rejected once the fabricated tissue is inside the body. I hope this is clear. Then moving on to the next element, we have growth factors and other bioactive elements, which are also added while the fabrication is going on. If I've heard of, there are a lot of growth factors like, as we have just discussed very briefly over hematopoietic stem cell, there is a growth factor called erythropoietin that is secreted by kidney, and erythropoietin helps in differentiation of hematopoietic stem cells into RBC particularly, not in other type of blood cells. Similarly, we have bone morphogenetic protein, BMP. There are different types of BMP. So if you put BMP in mesenchymal stem cell, BMP2 in mesenchymal stem cell, that will get converted to bone. If you put BMP7 on mesenchymal stem cell, it will be converted to cartilage. So there are different growth factors. There are different metallic elements that have a lot of role in differentiation of these things. Like magnesium is very important in development of bone cells. Calcium is equally important. Silicon is important. Copper is important in different cell types, different tissue types. So these are different elements that we must keep in mind while we proceed to design a tissue in the lab. Now, coming back to the first element that gives rise to scaffolds, and these are the biomaterials. So any material, whether it's synthetic or natural, whether it is harvested from a natural source, whether it is completely synthesized in the lab, any such material which can be used for healthcare purpose is generally termed as a biomaterial. And the polymers which are biomaterial are called biopolymers, as simple as that. We have a lot of examples where a lot of biomaterial and biopolymers have been used to give rise to completely synthetic heart valves, hip replacement. There are materials which are used in dentistry and different surgery. Just for example, the most common biomaterial, I'm not talking about biopolymers, starting with the broadly biomaterials, the most common biomaterial that you can think of is the stainless steel needle that goes inside your blood vessel to take out the blood sample during a pathological investigation. So the first property that must be there in the biomaterial is that it should not give any kind of irritation or any immunological response while inside the body. And that's why when the needle is going inside your blood vessel, you can experience the pain, but it's not causing any immunological reaction that can be problematic. So just again, going with the very basic definition, it can be any material which is natural or synthetic that can be used for non-interventional or interventional application in humans or animals for the medical purpose. Again, just trying to give a basic idea about intervention and non-intervention. Anything that is put inside the body is interventional approach. Anything that is applied over the body without disturbing the integrity of the skin is non-interventional. So if you apply an ointment over the skin, that is non-interventional approach. But if you are doing any surgery, that is an interventional approach. There are a lot of natural polymers such as chitosan, alginate, cellulose, carrageenan, collagen, gelatin, hyaluronic acid. These are all used in different applications for tissue engineering, depending upon what tissue we are targeting. There are a lot of synthetic polymers such as poly-L-lactic acid, polyl-glycolic acid, polyacrylic acid. There are a lot of bioceramics. There are other modified polymers. There are composite, which are made from integrating polymer and ceramic, which are used for different purposes. The basic property of any for choosing a biopolymer should be that it should mimic the natural extracellular matrix. If you can recall the structure of extracellular matrix, it contains a lot of proteoglycan. So proteoglycans are the modified polymer, naturally modified protein, where those proteins are attached to a lot of carbohydrate moieties. And those carbohydrate moieties are glycosaminoglycans, such as hyaluronic acid or heparan sulfate or chondroitin sulfate. So if we know the structure of those proteoglycans in the natural ECM, it will be easy for us to look for the biopolymer which can be very similar to those kind of ECM. The contents of the ECM, the proteoglycans, are almost common to all the organs, only their amount differs. And that's why the mechanical property of different organs are different. Like liver has a different kind of stiffness compared to kidney. Heart has a different kind of, heart and lungs are very expandable and contractile tissues. So they have different kind of mechanical property. And that's why we should always keep in mind the anatomy of the organ or tissue that we are targeting to replace with the natural materials. And that's why you always say while teaching this tissue engineering that the knowledge of anatomy and physiology is extremely important if you want to work with tissue engineering. There are a lot of different preparation methods or fabrication method for the scaffold. There are conventional methods and there are new methods, and out of those new methods, the most used method is called 3D printing or 3D bioprinting. So it is a very common term nowadays. I hope many of you have heard of 3D printing or 3D bioprinting. So a scaffold is a three-dimensional base structure that is fabricated of a suitable and biocompatible material that act as the substrate for engineering the tissue. As we have discussed, a scaffold should always mimic the natural extracellular matrix. It should have suitable porosity. It should be compatible. It should be compatible inside the body. It should be resorbable. It should be non-toxic as a whole. Its degradation product should also be non-toxic, non-immunogenic. It may be bioactive naturally and mechanically suitable for the target tissue. Okay. Again, just giving a brief introduction of resorption property. So we require the biopolymers to be resorbable inside the body, because if the biopolymer that is used to fabricate the ECM or the scaffold is not resorbable, it won't provide enough space for the natural tissue to grow. And therefore, the time of degradation of biopolymer must coincide with the time of formation of the native tissue over that space. Suppose a biopolymer degrades very slowly, but the natural tissue is forming at its normal pace. There will be a lot of ectopic tissue formation around the graft, and that will limit the mobility in the patient. That will be another problem for the patient. On the other hand, if suppose the polymer degrades very quickly, there won't be enough time for the natural tissue to grow and fill up that space. And there will be, again, a lot of problems because of that. There will be a huge void in that space. And therefore, the time of degradation or the rate of degradation of the polymer, the scaffold polymer, must coincide almost with the rate of growth of the natural tissue. Then coming back to the conventional fabrication techniques for a scaffold. So we have a solvent casting technique, we have freeze drying, which is called a lyophilization, and we have electrospinning. In terms of 3D printing or 3D bioprinting, we have certain techniques like fused deposition modeling, FDM, stereolithography, SLA, selective laser sintering, SLS, and there's a modified form of extrusion printing, which is called FRESH. We'll discuss a brief about these in coming slides. Okay. So if you can see the picture, if it is a bit clear to you, the first picture on the left-hand side corner top depicts the solvent casting technique, where a polymer is the a solution of a polymer is casted in in a required platform, and it is put into a temperature environment, mostly an incubator or a hot air oven, where the solvent evaporates, leaving behind a solid film. Most of the films that are created using this solvent casting methods are very flexible, and therefore solvent casting method was earlier employed to give rise to skin scaffolds, which are used for different types of wounds, superficial wounds, and second or third degree burn. Just coming down on the left hand side, you can see a technique which is called electrospinning, where the polymer solution is put into an instrument, from where it comes out. The polymer solution comes out from a nozzle in a very fine droplet type, and as soon as the droplet comes out, under the influence of electric field, the droplet is turned into a fiber. I hope you all have consumed cotton candy, and some part of the India it is called also called Hawa Mithai. So if you have seen the instrument that prepares the cotton candy, that is a typical simplest example of electrospinning. You put the solution of sugar and it comes out as a fiber. We use electrospinning a lot in tissue engineering, mostly for skin tissue engineering. In terms of drug delivery applications, electrospun mats are used a lot for drug delivery purposes. Just again coming down from electrospinning, you can see a scaffold structure which looks frozen. So in the first part, where we put the polymer solution into a tray, suppose the apparatus is a tray, and you put this polymer solution into a incubator, the solvent gradually evaporates, leaving behind a film kind of like a structure. If you put the same polymer solution into another type of vessel, and the instrument is called lyophilizer or a freeze dryer, then you will get much solid and porous scaffold, as you can see in the scanning electron microscope image. If you can see the porosity, it has got a very good porosity as per this image. Coming on to the right hand side, if you have seen how jalebi is prepared, so this is another analogy that I use a lot. Preparing a jalebi is the simplest, you can take it as the simplest example of an extrusion printer. So extrusion is a method whereby a thick viscous solution comes out of a nozzle either continuously or in a drop-wise manner. So you just put a pressure on the top, and the polymer solution comes as a thread. So jalebi preparation is the most common analogy for extrusion-based printer. So if you can see on the right-hand side, the extrusion-based printer is creating the scaffold with much uniform porosity and with a much better dimensional accuracy. And that's why three-dimensional printing or three-dimensional bioprinting has largely replaced a lot of almost all the conventional technique currently to prepare the scaffold or the tissue, in fact, not only the scaffold. With the conventional technique, it was mandatory, or it was we didn't have any other option but to prepare the scaffold separately and then seeding on the cells over that. But with the advent of 3D printing or 3D bioprinting, we just mix the polymer solution and the cells and the growth factors together. And this mixture of polymer, cells, and growth factor is called bio-ink. We'll discuss this technique in the coming slides. So the crux of this slide is that three-dimensional printing or three-dimensional bioprinting has largely replaced most of the conventional fabrication technique for scaffold preparation. Okay, now coming to the second element of a good functional tissue are the stem cells. So stem cell, as you might have known, are the basic undifferentiated cells that have two basic properties of self-renewal. That means they can proliferate without differentiation if the differentiation is not required, and they can give rise to a lot of cells depending upon the signaling that they receive. We have already discussed in terms of potency, a cell can be totipotent, pluripotent, multi or unipotent. Again, most of the time, as per other classification, we classify stem cell into embryonic or adult stem cell. As we have discussed, we harvest embryonic stem cell from the blastula stage of the embryo. However, in terms of adult stem cell, we can harvest it from any of the adult person through different locations. We already have discussed about iPSC. I hope you may recall that. Moving on to the next slide. So in terms of sources of stem cells from adult tissue, we have stem cell in the brain, we have stem cells in the epidermis, in the heart, liver, adipose, bone, and skeleton. They have their own type of stem cell present, so that in case of any minor injury, the stem cells themselves get those trigger, they differentiate into that particular type of cell and fill up that space. So a stem cell present in liver won't normally differentiate into a heart cell unless until they are deliberately triggered that response. So the resident stem cells, they have the one sole purpose of filling up the space if there are minor injury in that tissue or organ. And we have already discussed about the source of conventional embryonic stem cell and how do we reprogram a terminally differentiated cell back into the induced pluripotent stem cell. So what Dr. Shinya Yamanaka did through very conventional recombinant DNA technology, his team put a few transcription factors inside the skin cell. So he harvested the skin cell from the urine. So there are a lot of, in certain conditions, you can find epidermal cells in the urine, epithelial cells in the urine. He harvested the cells, transformed that with 4 transcription factor. One was OCT4, SOX9, c-MYC. These are the 3 most used transcription factor. Once they were inside the terminally differentiated cell, the cells give rise to embryonic stem cell like or pluripotent like characteristic, and these cells are induced pluripotent stem cell. Now these iPSCs can be differentiated to any kind of cell present in the body. Okay.